This section will help in identifying the anatomy for selecting the bone marrow aspiration site using the ileum surgical approach. The ileum may be approached posteriorly or anteriorly, with the posterior approach being suggested to minimize aspiration site morbidity. The posterior approach requires palpation to locate the superior aspect of the posterior iliac crest. The anterior approach requires palpation to locate the anterior superior iliac crest. First, prepare the surgical site and provide local anesthesia to the area unless general anesthesia is being utilized. Make a small oblique stab incision, a skin flap, just above the point of entry. This type of incision can easily be closed using a steri-strip or butterfly dressing following bone marrow aspiration. Remember, all marrow contact components must be rinsed with an appropriate concentration of heparin anticoagulant solution, including syringes and needle lumens and stylets, to prevent clotting. After identifying the anatomy and deciding on the needle trajectory, penetration of the ileum with the aspiration needle should be swift to avoid tissue factors coming in contact with the needle lumen and potentially resulting in activation of the clotting cascade. For an anterior or posterior approach, advance the needle by applying pressure simultaneously with back and forth rotation until a reduction in resistance is noticed, indicating entry into the soft marrow space. If there is difficulty in penetrating the ileum, simply tapping the aspiration needle handle with a mallet while holding the distal end of the needle at the point of insertion and at the appropriate angle should result in swift penetration of the ileum cortex. Penetration of the cortex will be noticed by a reduction of resistance as the needle enters the cancellous bone marrow space. For an anterior approach, introduce the needle through the prepared incision site, with the initial trajectory of the needle medially in line with the pelvic wing, as gauged by palpation of the inner and outer tables. The trajectory should be aimed slightly posteriorly at the iliac tubercle to enter the medullary canal just beneath it. For a posterior approach, introduce the needle through the prepared incision site with the initial trajectory of the needle, approximately 40 degrees lateral from the sagittal plane and 35 to 40 degrees inferior from the transverse plane. A general rule is to aim at the tip of the greater trochanter. Following penetration of the ileum with the aspiration needle, advance the needle distally into the marrow space 4 to 8 centimeters for the anterior or posterior approach, depending upon the volume of the ileum. Remove the stylet and attach a heparin-rinsed 20 milliliter syringe. Pull a small aspiration of 5 milliliters of marrow. Please note that short, quick aspirations of small volumes results in a greater recovery of nucleated cells than a long pull of a greater volume, which may result in dilution of the marrow with peripheral blood. If marrow does not aspirate easily, reposition the needle slightly. If marrow still does not aspirate easily, remove the syringe from the needle, remove the needle from the incision, and exchange the needle and stylet with a freshly heparin-rinsed needle and stylet. Reinsert the new needle into the incision site and penetrate the ileum approximately 1 to 2 centimeters from the original puncture site as we described earlier. If the first 5 ml of marrow is easily aspirated, the aspiration needle should then be rotated approximately 90 degrees, followed by another small aspiration of approximately 5 ml of marrow, so that a total of 10 ml of marrow is now in the syringe. Followed by retreating from the marrow space approximately 2 centimeters, and another small aspiration of approximately 5 milliliters of marrow is achieved. These steps are repeated so that a total of 30 ml of marrow is collected in the syringe. Care must be exercised to assure that the needle tip and side holes remain within the marrow space. If the needle tip and/or side hole or holes exit the marrow space, the needle must be exchanged with a freshly heparin-rinsed needle and stylet in order to prevent the likelihood of clotting of the harvested marrow. Remove the filled syringe containing 30 milliliters of marrow and immediately place it into the blood bag containing ACDA solution. Agitate or rock the bag so that the marrow is thoroughly anticoagulated. It is recommended to maintain the blood bag in motion as each volume of aspirate is added to the blood bag. Reinsert the stylet and advance the needle at a new trajectory along the same plane as the original insertion. The previous steps should be repeated, assuring that the needle tip and side holes remain in the marrow space. If the needle tip and/or side holes exit the marrow space, the needle must be exchanged with a freshly heparin-rinsed needle and stylet, followed by re-entry into the marrow space as described previously. Reinsertion of a freshly heparin-rinsed stylet addresses two important aspiration issues. First, the stylet will prevent the possibility of the needle from bending or kinking. And second, advancing the needle with a distal hole and stylet in place will prevent coring of the marrow and possible blockage of the distal and/or side holes of the aspiration needle. This last issue could also result in the activation of the clotting cascade and therefore can be prevented by following this recommendation. The previous steps should be repeated so that no more than 60 milliliters of marrow is harvested from a single puncture site. Again, the previous steps should be repeated with a freshly heparin-rinsed needle and stylet and a new penetration into the ileum made approximately 2 centimeters from the original puncture site, or the opposite ileum can be used if another 60 milliliters of marrow is required in 5 ml aliquots. Following bone marrow aspiration, place the marrow into the filter bag by attaching the filled syringe to the in-port connector on the filter bag. Inject the marrow into the filter bag containing ACD anticoagulant. Next, re-rinse the aspiration syringe with heparin solution as shown. And gently mix the marrow with the ACD. Continue to place aspirated marrow into the filter bag until the desired volume of marrow has been collected. If the aspiration needle is removed from the bone space and will be used to make a new entry, it must be re-rinsed with heparin solution so that any tissue or clotting factors are removed prior to making a new puncture. Re-rinse the aspiration needle as shown. Also, remove any dried blood and clotting factors on the surface of the needle by dampening a surgical sponge with the heparin solution and wiping the external surface of the needle as shown. Reassemble the sharp stylet and trocar so that it's ready for the next puncture. Once all the marrow has been harvested, the marrow filter bag should be inspected for signs of any clotting or bone spicules. Signs of excessive clotting may result in a poor recovery of nucleated cells. If seen, additional clot-free marrow should be harvested by closely following these collection guidelines. Next, open the slide clamp on the filter bag and then prime the filter by gently squeezing the filter chamber while holding the filter bag as shown. Attach a 60 milliliter transfer syringe to the outport of the filter. Then, while holding the filter bag above the filter, withdraw marrow by filling the syringe to the 60 ml mark. Once the 60 ml syringe is filled, disconnect it from the filter bag and attach a blunt fill needle to the male lure of the syringe.